Hello everyone, and welcome to a web series presented to you by Alpha MD. Today, we will be reviewing FDA's guidance for industry on determining the extent of safety data collection needed in late-stage pre-market and post-approval clinical investigations. Before we begin, for those of you who don't know us, Alpha MD is a strategic consulting company specializing in integrated services across pharmaceutical and healthcare organizations. Our service offerings range from data analytics, clinical trial operations, strategic risk management consulting, and telemedicine. Our versatile and experienced team comes from a variety of industry backgrounds while integrating principles within healthcare systems engineering and Lean Six Sigma disciplines and principles. Today, we will be reviewing the recent FDA guidance for industry that was published in February of 2016. This guidance is intended to help sponsors understand what selective safety data collection means, determine the types of safety data to collect, along with what types of clinical investigations may be appropriate, provide recommendations for when to consider a selective safety data collection strategy, and lastly, provide information to sponsors for consulting with relevant FDA review divisions to determine whether this selective approach would be considered appropriate. Our goal for this presentation is to provide a walkthrough of this guidance and identify who this guidance may affect, including the implications this may have on the design of your late-stage and/or post-approval clinical studies. Sponsors currently take a comprehensive approach to collecting extensive safety-related data points throughout drug development. However, in later stages of pre-market development or in post-approval studies, the guidance suggests that it may be more appropriate for sponsors to use a more selective approach in the safety data they collect for non-serious adverse events, those of which that have been well characterized through data in earlier stages. The guidance walks through a quick example that can be asked into a question. Ask yourself, if safety data collected on hundreds of patients indicate that 17% reported a headache when on drug treatment versus 10% that were on placebo, would an additional collection of this data be helpful in a larger phase 3 trial? Most likely not. We can determine that collecting the same data in thousands of additional patients in a larger phase 3 trial would minimally refine this value. In addition, this would require extensive resource utilization while providing no important new information. This is a great example that may lead a sponsor to a strategy for selective data collection. Selective data collection does not need to be limited to not collecting certain safety data. Sponsors may find that a reduction or less frequent collection of safety data may suffice. In addition, collection of certain safety data from only a fraction of the enrolled patients targeting a certain criteria can also be considered a good example of a strategy for selective safety data collection. If the drug safety profile has already been well established, selective safety data collection may be appropriate during late-stage pre-market and post-approval clinical investigations. To be more specific, let's take a look into the types of clinical investigations that may be considered for a selective safety data collection strategy. Selective data collection may be appropriate for clinical investigations, such as the 5 listed below. However, please note that they are not limited to just these 5. First, we have clinical investigations of new indications of approved drugs. Next, we have post-approval clinical studies conducted to fulfill post-marketing requirements and commitments. Third, late-stage pre-market and post-approval outcome trials. Next, we have pre-market clinical investigations for some original applications. And our 5th example of today would be post-approval clinical investigations in a different patient population or with different doses or other conditions of use. When it comes to clinical studies of new indications of already approved drugs, selective safety data collection may be appropriate if the existing safety database is relevant to the investigations. For example, the similarity of the population and doses studied in the new investigation to patients studied in the previous investigation. The data would be relevant in this comparison as it would be more of an apples-to-apples comparison. In post-approval studies that are being conducted to fulfill post-marketing requirements, the study population in these types of studies are generally the same or very similar to the population from which the pre-market safety database was derived. Therefore, safety data collection can often be limited to the primary safety endpoint and other endpoints of interest, while limiting or eliminating the collection of certain safety data that wouldn't help refine a value or be considered conclusive of great value to the safety profile of the drug. Outcome clinical trials usually involve relatively large populations, often anywhere between 5,000 to 15,000 patients, similar to populations previously studied. They are also usually longer in duration than earlier studies. Therefore, these studies typically have a fairly substantial safety database in existence before initiation of an outcome clinical trial, and this database already provides sufficiently precise estimates of common, non-serious adverse events. Even when there is not extensive prior safety data, a selective approach for this type of study could be collecting full data in only a subset of patients, while conducting a more selective data collection approach in other patients. Unless sufficient safety data already exists to adequately characterize the safety profile of a drug, comprehensive safety data collection is expected throughout pre-market clinical development trials intended to support approval of a new drug application or a biologics license application for a novel agent in order to elucidate occurrence, dose response, and subset variations such as demographics, concomitant illnesses, concomitant therapy, for the full range of adverse events of the drug. However, if sufficient comprehensive safety data becomes available before the completion of clinical development, selective safety data collection in some late-stage investigations may be appropriate. For example, selective safety data collection may be appropriate in phase 3 trials where the existing safety database adequately characterizes a drug's safety for more common, non-serious adverse events. Therefore, data on those events may not need to be collected. Think of the example that we went over in the introduction. There are also some cases of post-approval clinical investigations where either different patient population, different doses, or other conditions of use are being investigated, and they can be considered appropriate for certain elements when it comes to selective safety data collection. These elements would include the collection of common, non-serious adverse events in only a subset of the overall study population. An exception may also apply in cases where a lower dose or a shorter duration of therapy is being investigated. However, normally these types of post-approval investigations with differing patient populations or with different doses of use are usually not considered a match for selective safety data collection. Now let's take a look into the types of safety data that would be appropriate for selective collection. Where selective safety data collection is suitable, it may be appropriate to limit collection or data or stop collection of certain types of data, including the following. Since non-serious adverse events are well characterized when a drug is far along in development or has already obtained marketing approval, additional data collection would be unlikely to add to the established safety profile of a drug. For routine laboratory monitoring, it may be possible in some cases to eliminate routine laboratory monitoring or to decrease the frequency of monitoring certain laboratory parameters. Example, less frequent liver function testing as safety profile data diminishes concerns about hepatotoxicity. As long as the existing data satisfactorily characterizes all anticipated drug-to-drug interactions and metabolic pathways, additional detailed information on concomitant medications such as doses, dosing schedules, and start and stop dates may be of limited use, particularly for drugs used only for short term. Less detailed histories and less frequent physical exams for patients may be appropriate in some circumstances, especially in outcome clinical trials. If an abbreviated history is considered appropriate, but certain aspects of the history are considered relevant and should be collected, for example, cardiovascular disease, psychiatric history, including the history of depression, suicide, or both, these aspects should be described in the protocol. Sponsors should still always consider where there may be reasons to collect complete data to better characterize the safety profile of a drug. This may even apply even for the AEs that are not serious, fatal, or dose modifying, or that do not cause drug discontinuation or withdrawal from the trial. Two examples include specific population subsets and when there is a need to identify risk factors. If availability of data in a subset of a population is limited, generally all data on use of a drug in these specific populations should be collected, including data on non-serious adverse events. The incidence and severity of an adverse event may be related to baseline factors and other aspects of treatment. For this reason, where there is a known or suspected causal relationship between a drug and an adverse event, it may be important to continue collecting complete safety information throughout drug development to better characterize the relationship and the effect of these influences on risk. Sponsors should be aware that the following types of data are generally not appropriate for selective safety data collection and should always be collected. These include data on all serious adverse events, data on all non-serious adverse events that could lead to dose modification, drug discontinuation, or withdrawal from the trial. Also, data on unscheduled study visits, hospitalizations, and accidental injuries, because these events may reflect serious adverse events of the drug. In an oncology setting, data from all grade 3 and grade 4 adverse events, as well as grade 2 adverse events that affect vital organs. Example, the heart and liver. In developmental programs for rare disease indications, complete safety data should be collected because these trials generally have limited patient populations and are unlikely to meet the recommendations for selective safety data collection. It may be appropriate to collect certain safety data, for example, non-serious adverse events, from a sample of the study population. Methods for selecting the subpopulation should be detailed in the protocol. These may include selecting patients randomly, selecting study sites randomly, or selecting only the larger population sites. When selecting patient subsets or sites, it may be important to ensure representation from important demographic or disease-specific subgroups. Less frequent collection of a specific safety data, such as laboratory data and physical examination data, may be appropriate for long-term trials of drug with substantial short-term exposure. For example, laboratory studies and physical examination data may be collected at relatively long intervals, for example, every 6 months. Telephone and online follow-up between study visits may help maintain contact with patients. Regardless, specific safety data that will not be collected for the selective collection plan should be listed in the protocol. A sponsor considering selective safety data collection should consult with the relevant FDA review division or divisions to determine if such collection would be considered appropriate, and if so, develop its plan for implementation. Next, they should discuss its specified plan with the relevant FDA review division or divisions at the appropriate time. For example, at the end of a Phase 2 meeting for selective safety data collection that will be conducted in a Phase 3 trial. Lastly, they should incorporate the procedures for safety data collection in the study protocol, the monitoring plan, and other appropriate trial documents, including study functional plans. When asking who this guidance will affect, it is clear that all sponsors, CROs, and vendors will be affected. More specifically, those stakeholders who are developing protocols, study functional plans, and those who will be in discussions with the FDA on justifying their reason for a selective safety data collection strategy will also be primarily affected. We can assume a few implications of this guidance being issued. One would be the simplification of protocols and study functional plans, including the operational aspect of the data that would have originally needed to be collected and analyzed. We can also conclude that risk-based approaches are continuing to evolve, and the agency is providing efforts to highlight areas that may be appropriate. There is a direct effect on this guidance, helping to build efficiencies into the study conduct processes. For example, investigators, monitors, and auditors will be spending less time collecting and analyzing data that wouldn't improve the safety profile of the product. It's evident that risk-based approaches to conducting a clinical study have continued to evolve and have ever grown ever since the FDA published the risk-based monitoring guidance in 2013. Across the industry, unique strategies and approaches are being developed for how to best implement a risk-based monitoring strategy. The term risk-based has brought upon new meaning to other functional areas of clinical studies as well. Through many conferences, I've noticed that risk-based strategies are being established beyond just monitoring. One example is how sponsors and vendors are developing risk-based strategies for how they prepare for audits and inspections. I wanted to highlight this mainly because this guidance for selective safety data collection is no different. It is yet another way industry has stayed consistent in their messaging for approaches and strategies to become a more risk-based and more selective of what is most important and critical to the safety of patients and quality of data collected throughout a clinical study. I would like to thank you all for watching today's presentation. If you have any questions or would like to obtain a copy of the slides presented today, contact us and we will gladly answer questions and provide you a copy of the slides. In addition, if you have an idea for a web series topic or would like to be a guest speaker, please feel free to contact us as well. Again, I'd like to thank you all for your time today and have a wonderful day.